Hi, welcome to MTHFR support. Today we'll be going over the COMT and the MTHFR gene and how they work together. Medical disclaimer. You understand that any information is found within our website or this video is for general and educational and informational purposes only. You understand that such information is not intended nor otherwise implied to be medical advice. COMT, catechol methyltransferase. The relationship between the COMT gene, the MTHFR gene, and methylation is a fascinating area of nutritional genomics. Understanding how these genetic variations interact is key to navigating supplementation safely, especially for individuals with a slow COMT profile like COMT V158M and COMT H62H when they are homozygous. The COMT gene provides instructions for making the catechol methyltransferase COMT enzyme. Its primary job is to break down certain neurotransmitters, specifically dopamine, epinephrine, norepinephrine, as well as estrogen-related compounds. Think of it as the body's off switch for these stimulating chemicals. The specific variations we will talk about again is the COMT V158M homozygous, also known as MET-MET. This is how the slow COMT variant. It results in a 40% reduction in enzyme activity. Individuals with this profile tend to break down dopamine and adrenaline more slowly, leading to higher baseline levels of these stimulating neurotransmitters. COMT H62H homozygous, this variation is also found alongside the V158M and is also associated with reduced enzyme function. The enzyme's activity relies on helper molecules called cofactors. The most critical cofactor for the COMT enzyme is magnesium and its methyl donating partner S-adenosyl methionine. The enzyme transfers a methyl group from SAMe to its target molecule like dopamine to break it down. So SAMe availability directly regulates COMT speed. This is where the interaction with MTHFR becomes critical. The MTHFR enzyme is crucial for producing the active form of folate, 5-methyl tetrahydrofolate or 5-MTHF, which is necessary to convert homocysteine back into methionine, which then becomes SAMe. Then you have the slow COMT, V158M homozygous combined with MTHFR variations. It creates a specific biochemical environment. Research has shown that individuals with the slow COMT genotype who also have a genetic tendency for lower homocysteine, like MTHFR C677T when it's homozygous, can have lower baseline homocysteine levels. Here is why. The COMT reaction produces homocysteine. When the COMT enzyme works, it uses SAMe and produces S-adenosyl homocysteine, SAH, as a byproduct. SAH is then quickly broken down into homocysteine. A faster COMT enzyme would therefore generate more homocysteine. A slow COMT means less production when your COMT enzyme is running slowly. It uses less SAMe and therefore produces less SAH and subsequently less homocysteine. This can lead to homocysteine levels that drift lower than the optimal range. Why is low homocysteine a problem? While high homocysteine is a known risk factor for heart disease, very low homocysteine is also associated with health issues. including increased risk of neuropathy, inflammation, and certain types of cognitive decline. It indicates a possible imbalance in the methylation cycle, which is fundamental to hundreds of processes in the body, including DNA repair, detoxification, and neurotransmitter balance. This is why monitoring homocysteine is essential. You want it in a Goldilocks zone, not too high and not too low. For someone with slow COMT, standard methylating supplements like methylfolate and methylcobalamin can be problematic. Providing a large dose of preformed methyl groups can be like pouring gasoline on a fire, potentially leading to overstimulation, anxiety, and insomnia, because it may further overwhelm the already sluggish COMT enzyme's ability to maintain balance. This is why folinic acid, not to be confused with folic acid, and hydroxycobalamin, a form of B12, are often recommended as safer, more gentle alternatives. Folinic acid is a slow-release folate. Folinic acid is a naturally occurring form of folate that requires a few additional gentle enzymatic steps to be converted into the active 5-MTHF. This rate-limited conversion acts as a buffer, providing methyl groups at a slower, steadier pace that a slow COMT system can handle more easily. In contrast, direct 5-MTHF methylfolate floods the system with methyl groups all at once. Hydroxycobalamin is a reservoir form. Hydroxycobalamin is a natural form of B12 found in food. The body can convert it into the active methylcobalamin only as needed, effectively creating a reservoir. It also converts to adenosylcobalamin, the form used in the mitochondria for energy. This prevents the sudden spike in methyl groups that can occur with direct methylcobalamin supplements. If you are a homozygous COMT and also deficient in folate or B12, the methylation cycle can become underactive. A common strategy to kickstart the cycle involves using the active forms of these vitamins, but that leads back to the problem of overstimulation for slow COMT. This is where niacin, nicotinic acid, can be uniquely helpful. Niacin acts as a methyl sponge. To be cleared from the body, niacin requires and uses up methyl groups. Specifically, it is converted to trigonylene and N-methyl nicotinamide using SAMe. By doing so, it can rapidly lower an overload of methyl groups and consequently lower the levels of dopamine and adrenaline. Here's the scenario if you're deficient in folate and B12. Your methylation cycle is sluggish. If you then take niacin, you mop up the few methyl groups you have, potentially worsening the deficiency. The key is timing. The proposed strategy is to first use folinic acid and hydroxy B12 to gently build up your methyl reserves and improve methylation. If in this process you feel symptoms of overmethylation, anxiety, racing thoughts, irritability, taking a low dose of niacin, for example, 50 milligrams, can provide rapid relief by consuming the excess methyl groups that are causing the stimulation. To understand why someone with fast COMT usually does well with methylfolate and methyl B12, the COMT enzyme breaks down neurotransmitters like dopamine and epinephrine and uses S-adenosylmethionine as fuel, producing homocysteine as a byproduct. A fast COMT enzyme works very quickly. It consumes methyl groups from SAMe at a high rate and, as a result, produces more homocysteine than a slow enzyme would. This is supported by peer-reviewed research. A 2008 study titled Polymorphisms in Catechol-methyltransferase, the COMT gene, influenced plasma total homocysteine levels, found that individuals with the high activity COMT-BAL genotype had a significantly higher plasma homocysteine levels, particularly when they also had the MTHFR C677T variant that impairs folate metabolism. This is the crucial interaction. The MTHFR variation reduces the body's ability to recycle homocysteine back into the methionine and then into SAMe. So in a person with both fast COMT and MTHFR, you have two opposing forces that ultimately drive up homocysteine. Fast COMT is constantly producing homocysteine at a higher rate. MTHFR is less efficient at cleaning that homocysteine. Providing preformed methyl groups in the form of methylfolate and methylcobalamin is biologically logical. These supplements directly bypass the sluggish MTHFR enzyme, providing the fuel needed to convert the excess homocysteine back into methionine and thereby lowering it into a healthier range. A 2024 study published in Science Direct explicitly evaluated the effect of this exact combination, methylfolate, P5P, and methyl B12, on patients with MTHFR polymorphisms and confirmed its efficiency. For the fast COMT individual who is not prone to methyl group buildup, this is generally well tolerated and effective. Navigating this biochemical pathway is not a DIY project. And here is why working with a knowledgeable practitioner is so critical. At MTHFRsupport.org, we can assist with guiding you. Biochemical individuality, your specific combination of genes, including other SNPs not mentioned like MAOA or CVS, creates a unique blueprint. A practitioner can interpret these nuances. Dosing and tuning, finding the right dose of polinic acid hydroxy B12 or rescue niacin is an art. A practitioner can help you start low and go slow, finding the minimum effective dose and adjusting your protocol based on your feedback and lab results. They can help you distinguish between a good detox reaction and a dangerous overstimulation or imbalance. MTHFRsupport.org can be a valuable starting point for individuals and practitioners looking for community-driven information, educational materials, and potential guidance on navigating MTHFR and related genetic variations, including COMT. For anyone exploring this complex topic, it is always a good practice to combine information from advocacy and support organizations with peer-reviewed research and personalized care from a qualified healthcare practitioner. The interaction between genetics, biochemistry, and supplementation is highly individualized. We thank you for watching this video, and if you would need help with any of this, if you're a practitioner learning, or if you're a patient needing help on where to go when you're looking at all these SNPs, join us please at MTHFRsupport.org.